everyone, welcome to my YouTube channel. Today we are going to discuss about enzymes. This is the last topic along with the biosensors, where we use some of the enzymes for biosensors. We will be discussing in today's class. So previously we discussed about lipids. I hope you remember. If not, you can just go back to the previous video and check it. So let's begin. Enzymes. So usually enzymes are nothing but proteins only, but they have a separate function, specifically as they will act as a catalyst. Where they will act as catalyst? For in reactions, right? In chemistry we have seen catalyst, right? In a similar way, here enzymes will act as a catalyst in biological reactions. That's the difference. In biological reactions, whatever the catalysts are there, we can call them as enzymes. So they speed up the rate of reaction. So all of you know what do you mean by catalyst? They will speed up the rate of reaction. So here, without being consumed in the process, that means they doesn't undergo any chemical changes or chemical reactions, but they will accelerate, they will boost the reactions. So enzymes are specific to the type of reaction they catalyze, and they bind to the specific substrates to facilitate the reaction. That means, imagine there are 10 different biological reactions. So if, as I mentioned, different, so 10 different enzymes should be there as a catalyst. The enzyme which is acting catalyst for one particular reaction cannot, as another for another chemical reaction or biological reaction, it will not act as catalyst. That means it is highly specific to the particular biological reactions. So that is the basic and one of the important property of enzyme, its specificity. So enzymes play a crucial role in various metabolic pathways, digestion, and as well as cellular respiration. So metabolic pathways, all of you know, whatever when you eat the food, it will be in the form of starch, carbohydrate, or a protein, or whatever it is. That will be digested, right? So once that is digested, it should get from there, from starch, to convert it as energy. There are thousands of cycles, biochemical cycles which are going on inside our body. So that's where these enzymes play a very crucial role. They increase the rate of these kind of reaction, and even digestion and as well as cellular respiration. So let's see, in schematically, if you want to explain how does it acts. So you have a substrate here, and this is the enzyme. When you use the substrate, when you use the enzyme in the particular reaction, so enzyme substrate complex will form. So as I told you, enzyme is highly specific. In what way? It will be based on the shape. Most commonly, based on the shape only, it is highly specific. Substrate will fit into the enzyme, so that this particular reaction will take place and product will be produced faster. Understanding? You can see here the pinkish color substrate which is fitting in the enzyme, so forming enzyme substrate complex, and once the reaction completes, you will get the products like this. So what it says, enzyme is increasing the rate of reaction. So properties, as we discussed in the introduction, it's known for its specificity, reactivity, stability, renewability, and cost effectiveness. So engineering applications, bioremediations. So you know that enzymes are used to break down the pollutants in the environment. You can also use the enzymes if any pollutants usually will be in long chain. So you need to cut down. So that is called as a bioremediation. So these particular pollutants can be, when you cut down these long chain, they will become less toxic. So for example, such as oils, pesticides, toxic waste, all those things comes under the picture. So enzymes can be used as a bioremediation agent. Next, biofuel production. In biofuel productions also, like we discussed in the previous classes about lipids used in biofuel production and all. So enzymes are used to convert the plant material into biofuels. So when there is a reaction is going on where the plant material is converted into the biofuels, the enzymes will be added so that they will fasten the reaction, such as ethanol and biodiesel. So, food and beverage productions, enzymes are widely used in the food and beverage industry for processing, such as like baking, breeding, cheese making, juice production. In all these kind of methods, there are still many are there. Okay, so here in all these reactions, these are the reaction normally it will be happening, but if we add the enzymes, it will give the faster, it will fasten the reaction. Next, textile production, enzymes are used to remove the stains, whiten fabrics, and as well as to improve the softness of the textiles. So detergents, enzymes are used in laundry detergents to break down proteins, starch, and little stains. So pharmaceutical applications, it also has got pharmaceutical applications, such as it will be used in the production of various pharmaceutical products such as antibiotics and vaccines. We discussed about vaccines in the previous classes, DNA vaccine and RNA vaccine. Next, research and biotechnology. In every field, there is something called R&D, research and development. That means something which is coming, new ideas are coming in, and they are making new products or new engineering products or whatever. There it can find the application. Enzymes are used in tools in genetic engineering, protein engineering, and as well as molecular biology. So, biosensors is one of the subtopics under enzymes. So, biosensors, glucose biosensor, glucose oxidase biosensor is the topic. But in order to go to there, we have to just learn about what is biosensor. So, what are biosensors? Sensors, all of you know, a device which can detect something. Whereas biosensors will be having the analytes will be there, no? The element, recognition element. That will be biological recognition element. That is the only difference here. So that it will determine only biological entities. Understanding? So that's why for that we need biological recognition element. So biosensors are analytical devices that combine a biological recognition element with a transducer to detect and quantify target analytes. Now all of you know biological recognition element, just now I told you. So transducers, what do you mean by transducer? It's a device which converts one form of signal to another form of signal. That all of you, if you are coming from any engineering background, you will be knowing what is transducer, right? So along with this, we can use for detection and as well as quantifying target analytes. Detection is just a simply detecting. Quantifying is how much concentration of a particular biological entity is present in the given sample. That is also can be identified. Now the biological recognition element, that can be an enzyme here. Now the recognition element, what could be the recognition elements? Here it can be the enzyme, antibody, nucleic acid, or other biological molecule that specifically interacts with the target analytes. So as I told you, what is the difference between sensor and biosensor? Here recognition element will be biological entity. So what are the biological entities? Enzymes, antibody, nucleic acid, and other biological molecules. Understood? So now the transducer, what it will do? It will convert biological response, which is change in biological reaction in these particular enzymes, antibodies, and as well as nucleic acids or other biological molecule into an electrical signal that can be quantified and interpreted so that we can say that how much percentage of antibody is present in the given sample, or how much percentage of enzyme is present, or how much concentration of this particular nucleic acids are present. See, this is how the biosensors look like. You have analyte or a sample matrix. These are the samples which you want to detect, and biological recognition element. Here it can be, as discussed in the previous slides, antibody, enzyme, microorganism, DNA, cells, any biological molecule will act as analyte, so that it can determine. So here it is specific. For example, if I want to detect antigen, so what should be my biological recognition element? It should be antibody, because antigen and antibody complex will form, and then the biological response will be there, and this response will be detected by this particular transducer. It can be electrochemical, optical, mechanical, based on the what analyte is having the reaction. So readout technology and amplifiers will be there. Amplifier will amplify the given signal, and that will be used for the interpretation of the data by using data acquisition and processing. So biosensors have a wide range of applications in the form of medicines, environmental applications, or environmental monitoring and food safety. All these kind of major applications you can see. For example, biosensors can be used to monitor the blood glucose. So now this blood glucose levels in the patients with diabetes detects the contaminants in the water and food, and also monitors the environmental pollutants. So enzymes used in biosensors. So there are many enzymes which are being used in biosensors, but most commonly are glucose oxidase, lactose oxidase. So glucose oxidase, as the name itself indicates, it is used to check the blood glucose in the patient to detect especially the patients with the diabetes. Now the enzyme oxidizes the glucose, whatever the glucose will be there, no, it will oxidize that glucose to give gluconic acid and hydrogen peroxide, which is then detected by the transducers to quantify glucose levels in the body. So if the more the glucose, the more the concentration of the gluconic acid will be there. So based on that, it will quantify how much percentage of glucose is there in the diabetes patients. Then lactate oxidase. So it is used in determination of lactate levels in the biological fluids. Okay, if any biological fluids are there, if I want to determine how much lactate concentration it is there, that can be used. So biological fluids, what it can be the blood or urine, right? So when you go to a doctor, they will ask you the either the blood sample or urine sample to detect your lactate. Okay, so lactate oxidizes. What it does, see, glucose oxidizes. It was oxidizing glucose. So lactate oxidizes will oxidize the lactate. So there it was glucose to gluconic acid. Here it will be lactate to pyruvate, right? So lactate to pyruvate, which is then detected by the transducer. Next we are having cholinesterase. So cholinesterase is used in the detection of organophosphorus pesticides and nerve agents. So here we simply call it as HA. So this hydrolyzes acetylcholine. Means it hydrolyzes acetylcholine. So every function is giving, initially I am giving the definition followed by the function. Okay, what it does here, it is doing hydrolyzing the acetylcholine, and then the decrease the acetylcholine levels in levels will be detected by the transducer, so that they can quantify the presence of toxic substances. Next, alkaline phosphatase or ALP. It is also used in the detection of the inorganic phosphates. For example, it is these inorganic phosphates, where it will be present. It will be present in the water, which is wastewater or fertilizers. So the phosphates will be there. We need to check whether how pure this water is, or whether it is edible, or whether it can be drinkable, or whether it can be used for any purpose and all. So ALP catalyzes the hydrolysis of phosphates to produce signal that can be quantified. Next, the urease. Urea is used in the detection of urea levels in the biological fluids. How much urea is present in your biological fluids. Here usually for biological fluids to detect urea will be given as a urinole. So urease catalyzes the hydrolysis of urea. Once the hydrolysis of urea happens, it will produce the ammonia and carbon dioxide, which can be quantified by the transducer. So those are some of the examples of enzymes. So glucose oxidase in biosensors, again subtopic under enzymes. Glucose oxidase, here the enzyme used is glucose oxidase, and its application, how it is used in biosensors. It is an enzyme which is commonly used for biosensors for the detection of glucose levels in biological fluids such as blood and urine. Same point which is repeated from the previous slides. So the enzyme, this what it does, what's the function of this glucose oxidase? It catalyzes the oxidation of glucose. So once the oxidation of glucose happens, what products we will get? Gluconolactone and hydrogen peroxide, which can be easily detected and quantified by the transducer. Now in glucose biosensor, what happens? Glucose oxidase is typically immobilized on substrate. How it will be there? Immobilization is nothing but attaching on the surface. So for example, if this is the surface, we are attaching this particular enzyme on a substrate, okay, such as polymeric films to ensure stability and specificity. Now the transducers in the biosensors can be an electrode. or a fluorescent based system or any other type of sensor which is depending on the desired level of sensitivity and specificity. Based on the specificity and the sensitivity, we can use either components as transducers. Advantages: sensitivity, definitely it's very highly sensitive and can detect analytes at lower concentrations also, making them useful in applications that require precise quantification. Even we are having imagine 0.1 millimolar concentration of particular glucose or any gluconic acid or hydrogen peroxide, that can be detected by this particular glucose oxidase or by sensor. That's why highly sensitive. Next, specificity. As we told, enzymes are highly specific in nature. So these biosensors are working based on the in the presence of glucose oxidase enzymes. So they specifically recognize a target analyte which minimizes the interference, okay, from other substances in the sample. That means there will not be any disturbance. So it is highly specific to the particular reactions. So rapid response time. So biosensors can provide results in real time and making them useful in situations where quick results are required. Wherever you need quick results and real time data. Real time data is nothing but, it's not like you are given the sample and your results will come after 2 hours, you come and check, not like that. The moment you just use your sample, and that moment only the quantification should be there, detection should be there. So rapid response time is that. Next, portability. So since it is biosensors can be designed as a smaller devices which can be portable, making them useful in field applications, and also it can be carried to the remote locations also. You don't have to come to hospital every time. So cost effectiveness, biosensors can be manufactured at lower cost, making them an attractive alternative to more expensive analytic methods which are used in the laboratory for many applications. So limitations: stability, definitely. Biosensors can be affected by the environmental conditions. What are the environmental conditions? Some of the factors which can affect the stability of the biosensor are temperature, pH specifically, which can lead the degradation of the biological recognition element and loss of sensitivity. We know that biological recognition elements are nothing but biological molecules. We are using antibodies, we are using the enzymes, proteins, okay, bacteria. These all are highly sensitive to the temperature. If the temperature is high, what happens? They will degrade. That means they will get ruptured, or if the bacteria means higher temperature, they will get killed. If the antibodies are any protein molecules or biological molecules, they will degrade. Degrade in the sense they will just diffuse. They will lose their property. So that was about stability issue. That is first issue or limitation. Next, interferences. So biosensors can be affected by the other substances in the present sample. In the previous slide, when we discussed about interferences, we will not be there, but somehow in some cases we can find these interferences so that it can affect the performance of biosensor. Next, calibrations. So biosensor, any devices requires calibration. So does biosensor. That's it. Calibration, you know, all of you. Because if I am setting a particular device in the laboratory, I cannot use it for 5 years or more than 2 years. Okay, without calibration. Calibration is required every year or twice a year or kind of stuff. Why? Because to check whether this particular device is working properly or not. Next, complexity, limited shelf life. So biosensor has got a limited shelf life because biological recognition element may degrade over time. Imagine microbes I am using as a bioanalyte. So I cannot keep microbe alive for for years or months, right? So that's why the limited shelf life. Complexity. So biosensors can be a complex to manufacture and use, because here the metal parts are not only involved, because even the biological parts are required. So requiring specialized equipment and as well as expertise to operate effectively. So there is one more enzyme. So far we discussed about glucose oxidase. So there is one more enzyme, that is lignolytic enzyme in biobleaching. This is one of the application and subtopic under enzyme. Biobleaching, all of you know, is nothing but a process that uses biological agents such as enzymes. Why we use bleaching, all of you know, right? So similarly, here it is biobleaching. So it uses enzyme to remove the color and and it removes the colors or any stains which is present in the fiber, the distinct textiles, and brighten the fibers or paper or textiles. So it is a sustainable alternative to the traditional chemical bleaching methods. So whatever we are using right now in our household, it's 90% chemical bleaching methods. Okay, there is which contains harsh chemicals. such as hydrogen peroxide and chlorine. Okay, that can be avoided by using this lignolytic enzyme. Next, advantages: sustainability. Biobleaching uses biological agents such as enzymes, so they are renewable, biodegradable, and environmental friendly, and it will not affect any environment, any kind of environment. Even aquatic animals also will not be affected after cleaning, after releasing this particular dirt. Okay, so it will act as a very best thing compared to the traditional chemical bleaching methods. Next, improved product quality. So biobleaching can result in higher brightness. Okay, it can remove the color stains higher, effectively, so that more brightness will be there, and a more uniform color compared to the traditional chemical bleaching, leading to improved product quality. So that bleaching will be uniform, so that the color or texture of the material will not go wrong or will not be affected. So we can maintain good quality. Next, reduced energy consumption. So biobleaching typically requires lower energy input compared to the chemical bleaching, right? So that is the reason. So you can reduce the energy consumption and associated costs. Next, elimination of hazardous chemicals. Biobleaching eliminates the use of harsh chemicals, right? Hydrogen peroxide, chlorine, that can be avoided. Those are nothing but hydrogen peroxide, chlorine, so they are hazardous to our workers or environment. Next, lower production of harmful byproducts. Biobleaching reduces the formation of the harmful byproducts, right, such as dioxins. The harmful products will not be there as a byproduct. That can be produced during the traditional chemical bleaching methods. That can be avoided since there is no dioxins in biobleaching. There is no toxicity, and hence there is no harmful effect. Limitations of biobleaching: high cost of enzyme production, low efficiency compared to the chemical bleaching, need for further research, lack of widespread implementation. The first thing, high cost of enzyme production, definitely it requires a little bit extra effort, because as of now, whatever industrial scale we are having, all are chemical bleaching, right? We need to sometimes we may have to replace the devices or the big containers or which are used for these particular chemicals. Next, need for further research. It requires a lot of research efforts, so that to get the optimal biobleaching molecules or the products. Next, lack of widespread implementation. That's what, since the industry level, large scale industry level, it is not yet implemented, that need to be implemented. So next, lignolytic enzyme in biobleaching. Under this only we can again discuss same thing, lignolytic enzymes. For examples here, what we can give is lactases, peroxidases, and manganese peroxidases. These are some of the examples which are used in biobleaching to remove the color and brightens the fibers, paper, and textile. The same topic, line is repeating. These enzymes catalyze the oxidation of colored impurities in the fibers, resulting in a brighter and more uniform color. So lactases are copper-containing oxidases that catalyze the oxidation of lignin. So what is lignin? It's nothing but a complex polymer which is found in the plant cell walls, as well as other compounds such as phenols and aryl alcohols. Now, why you are talking about lignin here? So, because in industrial scale, the lignin has been widely used for many applications. In order to catalyze, in order to use for there, so we can use the lactases so that it can catalyze the oxidation of lignin. Next, peroxidases. These peroxidase enzymes are the enzyme that use hydrogen peroxide to oxidize organic compounds. Next example, we have manganese peroxidases. These are the enzymes which can use the hydrogen peroxide to oxidize lignin and other compounds. So now, lignolytic enzymes used in biobleaching are typically produced by the fungi or bacteria. See, lignolytic enzymes are produced by what? Fungi or bacteria, or immobilized on the support. Now you know what is immobilization: attachment on the support. Here, what can be the support? Here it can be the ceramic bed or a cellulosic matrix to ensure stability and prolonged activity. If you want to use this particular lignolytic enzyme for a long period of time, you should attach that on a particular substrate. Now the substrate can be here cellulosic matrix or ceramic bed, so that they will give you the stability and prolonged activity to the lignolytic enzymes. Now the immobilized enzymes are then added to the fibers. Now you have immobilized it. Now you add it to the fibers, where they catalyze the oxidation of colored impurities, resulting in a brighter and more uniform color. So that's it for today. If you have any doubt, please get back to me. Okay. Thank you, and this is the end of module 1.